Hello, everyone. So my name is Hong Tao Li from University of Southern California. It's a pleasure to be here to present our work about combination treatment of DNA hypomethylating agents with other anti-tumor drugs to target SETD2-deficient kidney cancer. I have nothing to disclose. So there are three parts about my presentation. First will be a brief background introduction. Then I will talk two works about our recent study. Okay, and we all know the epigenetic regulation can control gene expression without changing DNA sequence. So it includes, but not limited to, DNA methylation, histone modification, nucleosome occupancy, and the small non-coding RNA. So the epigenetic regulation plays a very important role for almost all cellular process, like cell differentiation. All the type of cells have a same genome, but the gene expression is totally different, and the function is unique. So this must be controlled by the epigenetic regulation. And this very strict and restricted. So if something happened and the epigenetic regulation changed, they will cause uncontrolled self-renewal of the cell. It will lead to the cancer. So this epigenetic alteration has been used as a markers for diagnosis, prognosis, and also be used for the therapeutic target. So the most important thing for the epigenetic regulation is all of these regulation factors are pharmacologically reversible. That means this change can be reversed very easily by small molecule inhibitor. And also, up to today, there are a lot of epigenetic drugs has been used for clinical usage. So our lab mainly focuses on the DNA methylation in cancer. So as we all know, in cancer cells, DNA hypermethylation usually happens as the promoter region of tumor suppressor genes and the silenced tumor suppressor genes. The hypomethylation usually happens in the repeat sequence, like a heterochromatin, which will lead to the genomic instability. Both of them will lead to the cancer. So besides this, our lab also have a work to report that the gene body hypermethylation at a subgroup of oncogenes could also activate gene expression. Then the silent the tumor suppressor genes by hypomethylation and the upregulation of the oncogene by gene body hypermethylation will cause to the cancer. And our work shows if we reverse the hypermethylation by the DNA hypomethylating agents like 5-ASA-CDR, it will silence oncogene and reactivate the tumor suppressor genes. That will inhibit, at least partially inhibit tumor growth. Okay, next I will introduce two commonly used hypomethylating agents, 5-ASA-cytidine and 5-ASA-deoxycytidine. We also call it DAC, and the 5-ASA-CDR. So this is the molecular structure. We can see the 5-ASA-CDR is a homolog of deoxycytidine. It can be incorporated into genomic DNA during DNA replication. After that, the DNA method transfers will recognize this 5-ASA-CDR, and but it cannot be methylated. The DNA methylation method transfers will be trapped by the 5-ASA-CDR and leads to the protein degradation. So that will cause the pharmacological knockdown of DNA methylation method transfers. So then the newly synthesized DNA will cannot be methylated. So after several rounds of replication, the whole genome methylation will be passively decreased. Also, because this molecule will exist on the genome for a long time, so that will cause a long-term anti-tumor memory. So this is here I will show one example from our work. We treated the colon cancer cell line HCT116 only for 24 hours. Day 0 shows the colony formation, show the cell growth. On day 5, we'll see the most inhibition of the cell growth. Then after that, we keep growing the cell, and gradually the cell growth rate will recover. Also on the right side, we'll see the DNA methylation after the treatment. On day 5, we'll see the most significant decrease of DNA methylation. Then some region, the DNA methylation will gradually rebound. So this region is what I talked about before for the hypomethylation and oncogene. This rebound methylation will reactivate the oncogene, so the tumor cell growth will gradually recover. Besides the DNA methylation, histone modification alteration also commonly happen in multiple cancer types. For example, the loss of H3K36 methylation. All these events are showed here will lead to the H3K36 methylation deficiency. So here in our work, we choose the ccRCC as a study model, which will have a highly mutated rate and SETD2 genes. SETD2 protein is the only well-known H3K36 trimethyl transfers in human cells. So the reason we choose the ccRCC as a study model is first because the 5-year survival rate will decrease dramatically if the tumor metastasized or become aggressive. In this figure, we can see even the SETD2 is not the most highly mutated gene in ccRCC, but when we talk about the metastatic ccRCC, the red bar here, we can see the SETD2 gene mutation is dramatically enriched. So this suggests the SETD2 mutation might be important for the ccRCC metastasis. And we check the survival rate using the TCGA patient data. We found the low SETD2 patient have a bad outcome. Okay, the first story I will be talking about is the synergistic inhibition of SETD2-deficient kidney cancer by the combination treatment of HMA and immunotherapy drug. We checked the immune response genes in the TCGA patient dataset, and we found the antiviral response possibly related genes are downregulated in SETD2 deficient cells. So that's come to one thing. It's called a cold tumor. So if the tumor have less immune suppressing cells, and they will have less immune response. So this tumor will have less response for tumor therapy. So we call it cold tumor. Based on our analysis, we think SETD2 deficient cells tumor might be a cold tumor. Then we'll try to find if there's some way we can turn the cold tumor to hot tumor, make the cell more sensitive to immune therapy. Also, the DNA methylation is changed in SETD2 deficient tumors. We see the hypermethylation and the upper panel region, and also the hypermethylation in the lower panel region. So this gives us a question: if we can reverse the DNA methylation by DNA hypomethylating agents and inhibit the cell growth, then we tested the sensitivity in 7 kidney cells, and these cells with different SETD2 status. So we use the H3K36 trimethylation as a marker to check the SETD2 status. 3 cell lines labeled by black have a Y-type SETD2 genes, and 2 cells have a homozygous mutation, the A704, A498. So they totally lost the histone modification. And the KK1 have a heterozygous mutation, SETD2, and the 769P is for some reason decreased the SETD2 expression. From the IC50 assay, we can see the 2 SETD2 homozygous mutation cell lines are very sensitive to DNA hypomethylating agents compared to the SETD2 Y-type tumor cell line. We also did knockout of SETD2 in 7860 cells, and we did the cell doubling time. The larger cell doubling time means the more inhibition here. So we can see for the knockout cells, we treated with DNA methylation, hypomethylation agent, the doubling time dramatically increased. That means they have most inhibition on this cell line. So this supports our hypothesis that the DNA hypomethylating agents could be targeted SETD2 deficient cells. Then we checked the GSEA pathway, and we found the interferon-related pathway are highly activated in the KO cell compared to the wild-type cells. So this result was confirmed in the different cell lines. From here, we can see the 4 SETD2 wild-type cells. The interferon gamma response pathway genes can be upregulated, but it's a very minimum. When we compare to the SETD2 knockout and the SETD2 mutation cell lines, this upregulation is more dramatically. We already know the DNA hypomethylating agents can upregulate transposon element and stimulate anti-tumor, antiviral mimicry. So what we check the T expression in the KO cells and the wild-type cells. We can see the DNA hypomethylating agents can upregulate the T expression in both of these 2 cell lines, but it's more dramatic in the KO cells. Then we check several histomodifications to find how this T is regulated. So the most dramatic change the histomodification is H3K9 trimethylation. From here, we can see even without the treatment, the D0, the knockout cells have more H3K9 trimethylation around this T region. After treatment, the H3K9 trimethylation level even further upregulated compared to the untreated cell. Then the people already know H3K9 trimethylation is associated with mRNA missplicing. Then we analyzed the mRNA missplicing, the intronic TE and the intergenic TE. So we can see the upregulated intronic TE is the major TE upregulated by treatment. Then we find several different type of missplicing. I will not to go to the detail due to the time limitation, but we further overexpressed 10 in vitro transcribed intron transcripts, which contain the TE region. So 8 of them can upregulate interferon pathway genes. So this supports our hypothesis that the missplicing induced the transposon element upregulation could stimulate the immune response. Further, we confirmed this in the mouse model. Because we want to test the immunotherapy combined with DNA methylation inhibitor. So we have to use the immune competent mice. So we did the SETD2 knockdown in the mice kidney cancer cell line. Both the RC50 assay and the colon information assay shows the SETD2 knockdowns mouse cell are all sensitive to DNA hypomethylating agent. Then we inject the cells into the mouse, the immune competent mice, and we did the epigenetic therapy 3 times a week, and plus the immunotherapy 2 times a week. After 4 weeks, we measure the tumor size, tumor weight. We can see the combination treatment significantly decreased the SETD2 knockdown cell tumor weight. And we also did the immune staining to show the CD8 T cell infiltration in the tumor. We can see in the SETD2 knockdown cells, the T cell have significant enrichment in the tumor. That means the immunotherapy cause more efficient in the SETD2 efficient cells. Then summarize this part work. We can see in all the tumor cells, the DNA hypomethylating agent can increase the T transposon element expression. But for the SETD deficient cells, because this inhibitor will cause higher missplicing. That further increase the double-strand RNA consequence in the tumor cells, will lead to more immune response. Okay, I'll move on to the second part about the increase of anti-tumor efficiency in SETD2-deficient kidney cancer by dual inhibition of DNA methylation and PARP activity. So people already published that the combination of a PARP inhibitor with a DNA demethylating agent caused enhancing the cytotoxicity, which targeting the AML model. So, but we wonder if this combination treatment also works on the solid tumor. And also, since SETD2 also functioning in the DNA repair pathway, and we also wonder if the SETD2 could be SETD2 deficient tumor could be a therapeutic target of this combination. Then we tested the combination treatment in 3 pairs of kidney cell line. Both have the SETD2 wild type cell and the SETD2 deficient cells. Yes, this supports our hypothesis, and we show even the single treatment of DNA hypomethylating agent or the PARP inhibitor all significantly decrease the SETD2 deficient cell growth compared to the SETD2 wild type cells. But when the combination treatment further inhibit SETD2 knockout SETD2 deficient cell growth, then we did the RNA sequencing and checked the pathway change, compare the 2 cell lines, the SETD2 knockout and the SETD2 wild type cell lines, and we found that the upregulated pathway are highly enriched in the immune response. But for the downregulated pathway, we immediately see the chromatin structure related pathway are downregulated, and also related to the DNA damage repair. Then we checked the double-stranded break marker gamma H2AX by the Western blot. We see the combination treatment can increase the gamma H2AX signal in wild type cells, but it's much higher in the knockout cells. That means the combination treatment cause more DNA double-strand break in the knockout cells. Then we checked the 2 DNA repair pathway, homologous recombination and the non-homologous joining. We see both these 2 DNA repair pathway decreased in the kill cells. That means the SETD2 kill cells have a DNA repair pathway deficiency. We also tested this combination treatment in the mice model. And here we see the relative tumor size. Both the DNA hypomethylating agent alone or the combination with the PARP inhibitor significantly decreased the tumor size. Although we didn't see the difference between the DNA hypomethylating agent alone or the combination treatment of the tumor size, but we do see the cell proliferation marker KR67 signal is significantly low in the kill cells after the combination treatment compared to the kill cells after the DAC treatment. That suggests if we optimize the mice treatment parameter, it might show the significant decrease of the tumor size. Okay, for the taking home message, first we see the SETD2 is a potential therapeutic target of DNA hypomethylating agent. Second, we see the hypomethylating agent could turn on tumor response and turn the cold tumor to hot tumor, make the tumor more sensitive to immunotherapy. Third one, we show the combination treatment can promote the therapy efficiency, which is very important to overcome the drug resistance. Because the drug resistance is the major reason for the therapy, for the fear of the therapy. So this might be show the potential new therapeutic strategy. The last one we show to identify specific alterations such as SETD2 mutations might be very important for the precision medicine. Yeah, we hope we can. In the future, we are planning to do the clinical trial to combine the DNA hypomethylating agent with immunotherapy. So right now we are looking for a collaborator and make these things happen. Okay, so we are thank our collaborator in our lab, also the collaborator from Hong Institute and the Sinai. Thank you for your listening, and I'd like to take any question. Thank you.